Hello, everyone. Thank you for tuning in to the Folding at Home Ask Me Anything. I am Bright Skies, moderator of the coronavirus in the Folding at Home Discord server. If you would like to join either of those, you can find invite links in the description.
So I'm Dr. Greg Bowman, and I'm an associate professor at Washington University School of Medicine and the director of the Folding at Home project.
For our first question, we have, how can folding at home help cure coronavirus when antivirals aren't related to protein folding?
Yeah, so folding at home is really far more general than protein folding. At a very fundamental level, what we're doing is asking how atoms in any molecular system push and pull on each other and move about relative to one another. And so as the name of the project suggests, the initial focus when it started 20 years ago was really on understanding this very basic research question of how do many proteins fold into these relatively specific shapes, which is a really cool process, right? It's as if you took all of the parts of a car, dumped them on your front yard, and they sprung together on their own without a mechanic or an engineer or anyone actively constructing the car, but they just sprung together onto their own into a functioning car. And if you saw this happen, you might say, whoa, how did that work? I want to understand that. And then maybe I could understand how to build a car, for example, or redesign the car. And so in the very early stages of the project, we're very much focused on this folding project, but given that, at base, the technology we're using is very broadly applicable, the focus has really shifted more and more towards understanding how the moving components of a protein contribute to its function and malfunction, and what new opportunities this opens up for designing therapeutics. And so that's really the angle here on COVID-19, is that we have the opportunity to provide increased levels of understanding of how the proteins that are involved in the virus's ability to infect human cells and suppress an immune response and make new copies or replicate the virus all perform these functions, and how we could potentially go about inhibiting those functions to combat the virus.
Thank you for the great answer to that one. Now, for our second question, why did you want to start folding at home?
Yeah. So for full disclosure, I should acknowledge that I didn't start folding at home. So my graduate mentor, Vijay Pandey, who was at Stanford University at the time, started the project while I was still in high school, I guess. And so I joined the project in 2006. He started it officially in the year 2000. So this fall will be our 20th anniversary. And in the intervening years, he's really shifted his attention to deep learning and ultimately to venture capital. And so, 2 years ago, he started bringing me in to be involved with leadership of the project. And about a year ago, I became the director of the project.
And our next question is, has folding at home done any good with SARS-CoV-2 research thus far?
Yeah, so we're still in the early stages ultimately. So the first project we started on folding at home related to COVID-19 and SARS-CoV-2 was launched about 6 weeks ago now. And so the fact that we have really interesting preliminary data is actually like an amazing rate of scientific progress. So we have started, I think, 100 projects on different, addressing different questions about proteins from the virus and how they function and how we can go about targeting them. And we're also screening over 50,000 small molecules to prioritize them for the likelihood that they bind tightly to the viral proteins and might serve as useful starting points for drug discovery. And so we can then go and test these experimentally and continue following up on any leads that work out well experimentally. So far from a computational perspective, I believe this week we'll pass off our first predictions about small molecules to our experimental collaborators to test. We've also been capturing some really dramatic structural changes that the spike undergoes. So this is a really cool system where you have 3 proteins arranged kind of in a circle, and the virus is able to protect itself from being recognized by the immune system by having these proteins close up on one another and cover over the conserved surface that it needs in order to bind tightly to a human host cell and initiate infection. And then the virus is free to change up the surface in order to keep our immune systems from finding patterns that they can recognize to detect when the virus is present. But in order for this strategy to work, this complex needs to be able to undergo this pretty dramatic opening motion to expose this conserved surface at least some of the time, so that when it's near a human cell, it has a chance of opening up and latching onto the human cell and initiating infection. And what the open state of this complex looks like and how the opening process occurs are both poorly understood. And so we're already making great headway with actually watching this process in our computer simulations and starting to extract structural information from that in order to help guide the design of new therapeutic strategies. And we're doing related things with other proteins from the virus that don't have such an obvious need as the spike does to undergo such structural changes, but where we expect to find interesting things like this based on the fact that almost every other protein that we've looked at, whether they're from Ebola virus or Alzheimer's disease or antibiotic-resistant infections, has had these really fascinating conformational changes that open up new avenues for the development of therapeutics.
And now, what do you envision Folding at Home to look like in 10 years? What projects appear to be most promising as of now?
Oh man, we have a lot of things going on in parallel. So prior to the emergence of the pandemic and our reallocation of basically all of our attention to this one set of related problems, my lab alone was working on Ebola virus and some proteins involved in various forms of cancer and antibiotic-resistant bacterial infections and Alzheimer's disease. So I think one of the really exciting things looking to the future is that what we're really developing is a platform for tackling biomedical research and human diseases. Diseases in general, if we wanted to, and so I'm looking forward to going back to applying this amazing computational resource to all of these other diseases that while less pressing immediately in the sense of relative to the current pandemic, are extremely important in their own right, as we also have something of an Alzheimer's pandemic looming in our future, for example, and serious issues with diseases like antibiotic-resistant infections and unfortunately plenty of other opportunities for pandemics with new versions of coronaviruses or Ebola viruses or you name it. There are a number of options.
For our next question, what is folding? Why does it matter? And is it anything like that mountain of laundry I can never truly overcome?
That's a good question. Yeah, there's a great quote from a writer, talking about protein folding and saying like, oh, it's not like, the neat piles of folded laundry that you create as you do your laundry. It's more like a garbled clothes hanger that doesn't have a recognizable shape. So these are really complex patterns, and it's really fascinating. This happens so reproducibly and spontaneously. So backing up, a protein is really a linear chain of chemicals called amino acids, and many proteins are these amino acids push and pull on each other in a way that they fold up into a very specific three-dimensional structure that confers them with the ability to perform some function. And these functions are quite diverse, so it can be sensing stimuli like in taste and smell, performing mechanical work like as in muscle contraction, or playing structural roles like in your hair. So they do all kinds of things. And so folding is really the process by which many proteins adopt this reasonably specific three-dimensional structure in a scientific sense. But in the sense of folding at home, we often just use the word folding to refer to participating in the project. And so more broadly, folding within the community just refers to participating in the process of helping us run these enormous simulations by contributing your personal computing power to the project. So one of the issues here is that experimentally we can often see what a protein usually looks like but we're blind to the moving parts that play essential roles in the protein's function. And if we could understand those, that might open up new opportunities into understanding how proteins function, how they malfunction, and how we can design new therapeutics. So by way of analogy, if you imagine you're trying to understand the game of football, and all you can see is what the players usually look like. Basically all you get to see is all the players in formation at the line of scrimmage. So there's a lot of valuable information there. How many players are there? Who's bigger, and maybe based on that, somewhat more likely to overcome the opposing player. But really, what you want to be able to do is watch the game unfold. And see how the rules come into play, and what the strategies for winning are, and how then you can get in there and manipulate the outcome by making different plays, for example. So that's what we're doing with Folding at Home, is we're taking these single snapshots from experiment that are extremely valuable, but are really just the tip of the iceberg, and adding in an understanding of what all the moving parts that contribute to their function and malfunction are. And then we can take that structural information and use it as a basis for things like rational drug discovery.
As a Folding at Home user, how can I best promote the software to others such that even less technically inclined people can understand the projects better?
Yeah, I think we've done a nice job. We have a number of students who have been very involved with social media and helping me work on blog posts on our website to try and explain in the simplest terms that we can what we're doing with the project. So I think if you're looking for material that helps explain our efforts, going to our blog at foldingathome.org/news is a great starting point. And one of my personal favorites, though of course I'm biased since I wrote most of it, is one that's what are we doing and how can you help in simple terms? And that's a great one for showing people to get them on board with what we're doing and bring them up to speed with the basics of the relevant assignments. And of course, then you're already on foldingathome.org and can switch over to the Start Folding link to download our software and start contributing.
As an Ubuntu user, is there a reason why the Folding at Home viewer software only supports long-term support releases?
So I'm going to back up and instead of addressing that specific question, to say that one of the great things is that we have a really vibrant community around Folding at Home, and this includes some great volunteers who help moderate our forum at foldingforum.org. And that's a great place to go and ask technical questions like this.
What kind of computing power is efficient enough to make a significant contribution to Folding at Home?
Yeah, so by design, part of the idea behind the project is that no individual computer can make a huge dent in the problems that we're tackling. Many of the easy problems, so to speak, from our perspective, would easily take 100 years on most people's computers if they tried to go it alone. And so part of our philosophy is to try to make participation in the project as broad as possible. So anyone with a computer can install and run the software and be a part of our efforts. So of course, with that said, the more computers we have and the more powerful they are, the better. So especially people who have invested in powerful gaming rigs or mining rigs for going after cryptocurrencies are able to make really valuable contributions to the project. But everyone's contribution is valued.
How can we identify fake Folding at Home websites or programs that will download viruses?
So with regards to security, we've actually always been very concerned about this and have numerous security features built in, like code signing, for example. And we've also recently had our software reviewed by Avast, one of the world's biggest antivirus software companies. And so they've given us a clean bill of health, and I hope to be able to tell you more about that in the future. But participating in Folding at Home is certainly as safe or safer than other things you might do on the internet. Yeah, to make sure you're at the right place, the best thing to do is make sure you're at foldingathome.org and our Start Folding or news pages.
My friends tell me Folding at Home is a bad idea. They say, You don't actually know what the program is doing on your computer. It could be mining for Bitcoin or something. What is a good way to prove that folding at home is legitimate?
Yeah, so I mean, we're really hard coded to run these molecular dynamic simulations, and in order to minimize security risks, that's our plan for the foreseeable future is to remain hard coded for that one particular task. And as I mentioned in a previous question, we have had external reviewers like our friends at Avast take a look at the code and confirm that it really is just running molecular dynamic simulations and returning that data. And you can always turn on the viewer to see what's going on.
Does folding at home really run well if you don't have an internet connection that is even bare minimum of normal daily use speeds? Also, does running this on an older spec laptop really help or not?
Yeah, so I mean, every little bit helps. Is what I would say in this case, and really these calculations are mostly compute bound. So I think most people's internet connections are such that the compute that they get done is worth more than, and the time invested in that is worth more than any minor delays from the time that it takes to upload the results.
Does folding at home utilize all of your resources even when you're just running a game in the background for grinding hours?
So that's really up to our volunteers. You can go in and toggle how much of your computer we're using, and you can elect to let us run all the time or only when your machine is idle so that we're never in your way.
How will profit generated from folding at home products be distributed? Who will gain from them?
Yeah, so Folding at Home is a public project that's funded mostly by federal grants. And so we try to keep everything that we're doing in the public domain. So in the past, we've made all of our data available upon request. And in the present circumstances, given the importance of the COVID-19 work and trying to contribute to accelerating a response to the pandemic as much as we can, we're working on ways to make all of the data that we generate on COVID-19 publicly available to anyone who wants it on the internet. And we're also exploring licenses that will help ensure that the data stays in the public domain.
How does folding at home compete with other distributed computing projects?
So Folding at Home is quite large at this point. We've got over 3 million devices that are running the software and contributing to what in aggregate is one of the most powerful single compute resources in the world. Even so, I mean, we're still just a small fraction of the compute power on the face of the globe. And so there's plenty of room for other distributed computing projects as well. And so our focus is really on understanding protein dynamics and implications of that for functioning, malfunction, and the design of therapeutics. Other relevant ones include Reusable, which is really focused on predicting the structures that one would observe experimentally, for example. So they're not really focusing on the protein dynamics that we're primarily interested in. And they also work on designing sequences to adopt those specific structures. And looking ahead, we'd be very interested in designing sequences, protein sequences to control protein dynamics. But in both cases, there's a lot of nice synergies between the projects rather than any sort of direct competition.
What issues do you foresee during future development, and what's the next big feature to be implemented that really excites you?
That is a great question. This is a challenging one, I would say the focus of federal funding has very much shifted towards more translational efforts. And so there's some nice efforts underway to fund infrastructure development, but some of the mechanisms that would have allowed us to get large amounts of funding to have multiple software developers working on Folding at Home, for example, are no longer available to us, at least in the obviously applicable form that they were before. So one of the challenges for us now is to figure out how to fund the basic research and infrastructure that we do that enables us to do all of the more translational work that we also have underway. But there's a lot of exciting things that we have the opportunity to do. So some of you might have noticed that sometimes the traffic is so intense that we have troubles keeping up with all the number of volunteers and making sure that there's enough work units for everyone to do. Part of this has to do with the architecture of FoldingAtHome and the fact that much of it predates modern advances in cloud architecture and cloud services. So we're actively engaging with a number of companies and organizations to come up to speed there and better leverage some of the technology that's out there. So we'd love to have people be able to upload data to buckets online, for example, in a way that can be done with a lot more parallelism than our current architecture. And that could really allow us to quickly scale up the number of projects that we can serve out to our volunteers at any given time. So that's on the more technical side, but on the user engagement side, we're also really interested in overhauling the client and having a system where our volunteers could log into what would essentially be like a Facebook profile and get in to analyze their stats more readily, and where we could have more interaction with people and update the UI more quickly to provide more up-to-date information on what we're doing, for example.
Are FoldingAtHome results reusable, or do you have to run a new project every time you have a new question?
Yeah, so the results that we generate are very much reusable. So oftentimes when we start a project, we do have a specific question in mind, but the proteins are doing what they want to do. And so it's very common that we go back to our data sets and start asking other questions that we never conceived of when we first started the project. And this is really nice. I mean, my wife is a developmental biologist and has done a lot of work with novel organisms like mice and other and cells and such. And it's great to work with data where you can have backups and put it into archives to dig out later without having to do a lot of restarting mouse colonies, for example, or waiting for mice to grow.
Are there any other ways to engage with the FoldingAtHome community?
Yeah, there's a whole plethora of ways. So there's a couple of things that we run. So of course there's foldingathome.org. We're also very active on Twitter, so you can follow our @FoldingAtHome account. And we've recently started up a FoldingAtHome Discord server, and you can find information on how to join that by going to foldingathome.org and then clicking to our COVID-19 page, and there will be a link to join. But one of the really cool things is not just the things that we're doing, the content that we're generating to explain what we're doing and the results that we're getting. There's a whole community that has organically grown up around FoldingAtHome, and so there's all kinds of websites that people have contributed to help track the points that people earn in proportion to their compute resources they contribute to the project, for example, and sites that people have made analyzing different hardware setups to figure out what gets you the most points or what hardware is the best points per dollar, for example. So there's a lot of resources that you can find by going and searching around a little bit.
What can we do with the points we earn?
Yeah, so right now the points are really a way of tracking what people's contribution is, and a way of having some friendly competition and gamification of the project. So there's nothing that you can exchange them for or anything like that right now, at least officially through FoldingAtHome. So we don't support any of these or things, or especially I should say we don't have any direct affiliations with any of these projects, but there are people that do things with the points, like use them as the basis for the distribution of cryptocurrencies. And looking into the future, we're very interested in exploring more ways of gamifying FoldingAtHome. So, we're still in the brainstorming phases, but I think it would be fun if people could take the competition a little further and make wagers about, okay, I'll bet you I can earn more points than you in the next 24 hours, and whoever gets more points also gets the other points, or something along those lines to make it a little bit more interactive. But we're in the very early stages of thinking about this and trying to get the resources to put software development effort into projects like that.
Is FoldingAtHome collecting my data?
No, we are not collecting our users' data, and in fact, we do so little data collection that it's hard for us to give precise numbers on how many people are participating at any given point in time.
Who will benefit from the results of the FoldingAtHome work units generated?
Yeah, so this is really a community effort, and we hope that the scientific community will be one of the initial benefactors in terms of our ability to uncover new scientific insights and share that with others. But more and more, we've moved into biomedical problems with direct translational benefit. So, recently we've discovered new ways to potentially combat Ebola virus and experimentally confirmed that we can potentially have real impact there. We've also found our first small molecules for potentially combating antibiotic-resistant infections. So things are really moving along in that direction and greatly expand the breadth of communities that we could potentially impact with our work in a very direct way.
With the current political situation in the US, will FoldingAtHome results only be made available to US scientists, or will others worldwide have access to it too?
Oh no, we're very interested in being fully in the public domain on a global scale. So we have participants in the project from around the world. It's really cool. In the past we've been able to generate maps where we show a little pinprick of light everywhere someone is participating in FoldingAtHome. And you can see that everywhere that's habited, there are people contributing in the project. And one of my students is working on updating this, and he is super delighted to see that even in Madagascar there are people running FoldingAtHome, so we're all over the place and have every intention of making the data available to anyone with an internet connection.
Where can I learn more details about what FoldingAtHome does?
Yeah, there's a lot of information on foldingathome.org, and you can also follow our Twitter account, which is FoldingAtHome, or my account, Dr. Greg Bowman. And we're doing a lot trying to explain both the basic science that motivates what we're doing and the projects that we're starting up and the results that we're getting in.
How do you compare to the world's largest supercomputers like the ones IBM has?
Oh man, this is one of the really exciting developments since we launched our COVID-19 work. At the beginning of March, end of February, we had 30,000 active volunteers in the project, which was great because that already enabled us to do a lot of science that we wouldn't have otherwise been able to do. But since we launched this project, we've had tremendous growth, and as I think I mentioned earlier, we now have over 3 million devices participating in FoldingAtHome. So according to our best estimates right now, we've broken into the exascale, and we're the first to do that. So usually the performance of supercomputers is measured in petaflops, and we're now measuring ours in exaflops. So another 3 orders of magnitude higher than a single petaflop. And it's estimated that the raw compute power of FoldingAtHome is more than the top 100 supercomputers combined, or something like that, which is a truly astounding resource to be able to bring to bear on the current pandemic and hopefully future health issues in the future. This is one of the things that I really want to emphasize, that every time I get the chance, is that we only had the opportunity to pounce on the current situation and start making a contribution because we already had this vibrant user base. And I hope lots of the people that have joined to help with our COVID-19 efforts will recognize that this really is a general platform that can add value in many scenarios, and that many of them will continue participating in FoldingAtHome afterwards and help us as we hopefully in the not too distant future have the opportunity to shift more of our focus back to Alzheimer's disease and cancer and antibiotic resistance and Ebola and these other human health issues.
How do I donate a server if I want to help?
So there, In the past, we've run the servers that are managed Folding at Home at the universities where the labs that participate in the project operate. More recently, we've been working with some companies to get servers running in the cloud to meet the current demand for work units, if you will, and engage with the most volunteers that we possibly can. If there's people who are interested in running servers, you can reach out to us through the Twitter account is probably the best way.
How can we see the results of those participating?
Yeah, so there's a variety of different ways to see what we're doing scientifically with Folding at Home. So if you really want to get into the details, you can go look at the publications that we're putting out in scientific journals on a regular basis. And if you are not interested in delving into the scientific details, we're always working to provide more accessible synopses of what we're doing through the project descriptions that show up in the software on your personal computer when you're running a project, and also through posts to our blog to try to explain in more lay terms what we've done when we publish papers, for example. We're doing the same thing through our Twitter account, so foldingathome.org and the Folding at Home account on Twitter are two really good resources.
I love folding at home, but I don't know how to explain it to my parents, relatives, or friends who are skeptical and unfamiliar. What are good points I can say?
Oh man, well I hope we've given you a bunch of good material through all of these questions, but I think I would say that in some ways drug design is a really empirical process that's much like a high stakes game of pin the tail on the donkey, because you're doing a lot of trial and error and just seeing what sticks, so to speak. And Folding at Home is a way where you can contribute to your computing power to a larger effort that's essentially a strategy for opening our eyes and having a better sense of how the world works and how we can get in and manipulate it to do things like treat diseases.
Thank you, everyone, for listening to the Folding at Home Ask Me Anything. Keep in mind you can get links to the website, Twitter, forums, and Discord server for Folding at Home in the description. You can also find a link to the coronavirus Discord server there too if you want to join. I hope you all enjoyed it. Thank you.
Yeah, I agree. Thank you very much, and thanks for hosting this, and especially thank you to those of you that are already participating in Folding at Home, and for those of you who aren't, I hope you'll join us up with us and help us to bring more compute power to bear on these important research questions.